If you look at the longevity space today, you are met with a wall of contradictory advice. One week, you are told to prioritize specific supplements. The next, you are told to adopt a restrictive diet. It is difficult to identify which interventions actually result in measurable cellular changes. When geneticists look at the biology of aging, they focus on a select group of master regulatory nodes. Among these, the gene FOXO3, or forkhead box O3, is the most consistently validated longevity factor identified across diverse human populations. Specific protective variants of the FOXO3 gene are found at a high frequency in the centenarians of Okinawa, Japan. Genetic association studies have identified those same longevity-linked alleles in Ashkenazi Jewish populations living in the United States and Europe. We find the same genetic correlation again in the Mediterranean centenarians of Sardinia, Italy. Research suggests that FOXO3 activity is not merely a marker of a long life, but the driver of the biological mechanisms that delay its decline. When activated, this gene executes a program that clears cellular damage and maintains genomic integrity. To control FOXO3, you have to understand a fundamental biological trade-off. Your cells cannot prioritize growth and repair at the same time. During nutrient abundance, high insulin keeps FOXO3 sequestered in the cytoplasm, inactive and away from DNA. When nutrients drop, AMPK activates, allowing FOXO3 to enter the nucleus. Inside, it drives cytoprotective genes, increasing antioxidants like superoxide dismutase and repairing DNA. This leaves us with a practical dilemma. If we want to initiate these repair programs, we have to trigger a state of biological stress. The question is how to do that without resorting to severe starvation or excessive physical strain. We can begin by analyzing the return on investment for the most common FOXO3 activators. By looking at the clinical data, we can find the most efficient balance between effort, cost, and the resulting biological activation. Fasting is the most accessible environmental intervention for extending health span. By temporarily lowering caloric intake, you reduce the insulin and IGF-1 levels that normally keep FOXO3 suppressed. You do not necessarily need multi-day fasts to see results. A study using animal models compared daily 16-8 intermittent fasting for 16 hours and eating for 8, against a prolonged 40-hour fast. The 16-8 schedule resulted in a 2.5-fold increase in FOXO3 expression compared to the 40-hour group. Human clinical trials have mirrored this, showing that cyclic fasting for 8 weeks significantly elevates both FOXO3 expression and cellular garbage disposal activity. This suggests a Goldilocks zone for fasting. Moderate cyclic stress appears to provide a sufficient signal to upregulate survival genes without triggering the severe catabolic suppression seen in deeper starvation. The trade-off is a requirement for daily discipline rather than a financial investment. This approach avoids the muscle loss associated with long-term calorie deprivation while still engaging the repair pathway. This positions 16-8 fasting as a baseline strategy, an intervention that increases gene expression without a financial barrier to entry. Physical exercise serves as a potent trigger for the AMPK pathway. As your muscles consume ATP during exertion, the resulting increase in cellular energy demand activates the sensors that move FOXO3 into the nucleus. Inside the muscle, FOXO3 transcribes genes that control autophagy. This visualization shows the process of mitophagy, where cells identify and clear out damaged mitochondria that have become dysfunctional during the stress of contraction. The benefits extend to the brain. Muscle contraction stimulates the release of irisin, which promotes the accumulation of FOXO3 in neurons. This muscle-to-brain axis has been linked to improved cognitive resilience and protection against oxidative damage. However, if the stress is chronic and lacks recovery, FOXO3 can shift from promoting repair to initiating muscle atrophy. Maintaining the balance between acute exertion and rest is required to keep the pathway in a survival-oriented state. Exercise acts as a physiological multiplier. It creates the acute stress necessary to accelerate cellular turnover, provided the volume does not exceed your body's ability to recover. Plants produce specific chemicals to protect themselves from UV radiation and drought. When we consume these xenohermetic compounds, they trick our systems into activating the same stress resistance networks, including FOXO3. The traditional Okinawan diet is rich in these compounds. Their reliance on the purple sweet potato, which is high in anthocyanins, provides a consistent dietary source of phytochemicals that interact with the FOXO3 pathway. Today, these compounds are available in concentrated supplemental forms, such as high-potency astaxanthin, which can cross the blood-brain barrier. In animal models, specific astaxanthin compounds have been shown to increase FOXO3 activation in heart tissue by approximately 90%. This is an exceptionally high degree of upregulation for a single dietary compound. Similarly, curcumin has been shown to restore function in aging stem cells by inducing FOXO3-mediated autophagy. This allows cells to clear the internal debris that typically leads to senescence. The trade-offs for these shortcuts are financial. High-quality extracts can be expensive, and their effectiveness is often limited by poor bioavailability. Your body may not absorb enough of the compound to reach the necessary concentration in the tissue. These compounds act as powerful amplifiers. While they can enhance the magnitude of your cellular response, they're designed to supplement, not replace, the foundational signals of fasting and physical activity. Environmental temperature stress represents a more extreme form of hormetic challenge. Deliberate heat exposure has been used for centuries, but modern data suggests its impact on longevity is tied to how our cells manage protein damage. Heat stress engages the heat shock response. This process involves proteins that work in tandem with FOXO3 to repair DNA and ensure that the proteins inside your cells remain properly folded and functional. Observation data from large-scale studies shows that frequent sauna use, 4 to 7 times per week at temperatures between 80 and 100 degrees Celsius, correlates with a 50% reduction in cardiovascular death risk. The difficulty lies in the commitment. This protocol requires specialized equipment and significant time, making it one of the most demanding interventions in terms of lifestyle adjustment. For those with the resources, sauna therapy is an effective lifestyle commitment. However, because of the high barrier to entry, it is considered a secondary intervention rather than a mandatory requirement for longevity. Now we can synthesize these findings to help you decide which strategies fit your own life. We can map these interventions on a matrix that compares the biological impact of the FOXO3 activation against the required effort and financial cost. Low-effort habits like drinking green tea for EGCG sit in the bottom left. They provide a moderate background signal for FOXO3-mediated tumor suppression and cell cycle arrest, with very little cost. Based on the 2.5-fold increase in mRNA expression seen in cyclic fasting studies, 16-8 intermittent fasting sits in the top left quadrant. This offers a high biological impact for almost no financial investment. The maximum impact strategies cluster in the top right. Combining the AMPK activation from exercise with the documented 90% gene activation from compounds like astaxanthin pushes the system to its highest measured output. The matrix shows that you do not need an expensive regimen to see results. The most accessible free habits in the top left quadrant provide a biological response that rivals the most high-cost interventions. For those seeking a pragmatic approach, combining a 16-8 fasting window with regular exercise provides a significant biological return for $0. It addresses the primary regulators of FOXO3 without complicating your daily routine. For those prioritizing maximum results, the focus should be on synergy. Adding targeted compounds like curcumin to a physical training program can help push the biological response further, protecting against inflammation during intense exertion. However, the biology of FOXO3 carries a clear limit. More activation is not always beneficial. Chronic 24/7 activation of this pathway through continuous fasting or overtraining can backfire. If the repair signal never turns off, it can lead to cell death and muscle wasting as the body consumes its own healthy tissue. The common factor observed in centenarian populations is metabolic flexibility, the ability to move between states of nutrient abundance and nutrient scarcity. Intentionally cycling between brief periods of biological stress and recovery reflects the metabolic patterns observed in the world's longest-lived populations. This remains the most evidence-based approach for utilizing the protective power of FOXO3.